Welcome back to the University of Melbourne Biology series. This is episode 10, and today we tackle one of the most satisfying corners of genetics, how genes actually behave together. So far you may picture each gene as a tidy switch, sorted at random and acting alone. The reality is richer. Some genes travel together because they sit side by side on a chromosome. Some genes team up, several at once, to shape a single trait. And every gene is held under tight control, switched on and off so the right product appears at the right moment. Over the next 12 minutes we will move through three connected ideas: how linked genes are inherited and mapped, how multiple genes interact to build one phenotype, and how cells regulate gene expression from start to finish. Let us begin. Start with position. Linked genes are genes located on the same chromosome, and because of that shared address they tend to be inherited together. They do not assort independently the way genes on different chromosomes do. But inheritance is not perfectly rigid. During meiosis, crossing over can occur between homologous chromosomes, exchanging genetic material between them. That exchange shuffles the deck, creating new allele combinations that did not exist on either parental chromosome. Here is the elegant part. The frequency of recombination between two linked genes is a proxy for the distance between them. Genes close together are rarely separated by a crossover, so recombination is low. Genes far apart are separated often, so recombination is high. Distance, written directly into how often alleles get reshuffled. If recombination frequency tracks distance, we can turn it into a ruler. That ruler is the genetic map. A genetic map is built by analyzing the frequency of recombination between linked genes, and it gives a visual representation of the relative positions of those genes along a chromosome. The key quantity is map distance, a measure of the distance between genes on a chromosome, calculated directly from recombination frequency. Map distance is more than a label. It carries predictive power. Once you know how far apart two genes lie, you can use that distance to estimate how often they will recombine, and therefore to predict the outcomes of genetic crosses. So a few careful measurements of recombination become a map, and that map becomes a forecasting tool. Let us put that map to work. The outcomes of genetic crosses can be predicted using genetic maps. By analyzing the map distances between genes, you can estimate the likelihood of recombination events and, from that, the resulting genotypes of the offspring. Recombination frequency is the engine of the prediction. It tells you how likely a recombination event is between the two genes you care about. This is why mapping matters so much in practice. Predicting cross outcomes is essential for understanding genetic inheritance, because it lets us move from observed data to expected results and back again. Measure recombination, build the map, read off the distance, and you can say what the next generation is likely to look like before a single cross is performed. Now we change the question entirely. Instead of asking how genes are positioned, we ask how they cooperate. Genetic interactions occur when different genes influence the same trait, and the result is more complex outcomes in genetic crosses than a single gene would give. There are two broad flavors of this teamwork. In an additive interaction, genes independently contribute to the phenotype, each adding its own effect. In an epistatic interaction, the expression of one gene is dependent on the presence of another gene. Notice how different those are. Additive genes work side by side, epistatic genes work in sequence, one depending on another. Both, though, share the same headline idea: a single trait can be shaped by more than one gene at once. Let us take each flavor in turn. Take the additive case first. Additive traits are characterized by the independent contributions of different genes to a phenotype, contributions that combine to produce a new trait. The genes are not competing or overriding one another, they are simply adding up. A clear example is flower color, where dominant traits from separate genes can combine to produce a new phenotype that neither gene would yield on its own. That is the signature of an additive interaction: bring the right dominant contributions together, and something genuinely new emerges from the sum. So when you see a fresh phenotype appear only when two genes both contribute, think additive combination, each gene quietly adding its part to the whole. Additive contributions are not the only way genes shade a trait. Some genes act as modifiers. A modifier gene is one that influences the expression of other genes, adjusting how a trait turns out rather than creating it from scratch. A familiar arena for this is coat color. Modifier genes can affect traits such as pigment distribution, and in coat color specifically, modifier genes can determine the type and distribution of pigment granules. So one gene might decide what pigment exists, while a modifier decides how that pigment is laid down across the animal. The modifier never works alone, its whole job is to tune the output of another gene. Keep that idea of dependence in mind, because it leads us straight into something stronger. That stronger form of dependence is epistasis. Epistasis is a gene interaction in which the expression of one gene is dependent on the presence of another gene. It is not a gentle tuning, one gene can effectively gate another. Because of that gating, epistasis can lead to complex patterns of inheritance, where the phenotype you observe does not map neatly onto a simple single-gene expectation. You can see epistasis at work in traits such as coat color in dogs and cats, where the action of one gene determines whether a second gene's effect is even visible. The lesson is that genes are not always independent voices. Sometimes one gene must speak first before another can be heard at all, and that hierarchy reshapes the inheritance we see. These interactions are not abstract, they build real bodies. Consider wing development in the fruit fly, Drosophila. The development of Drosophila wings involves complex gene interactions, and at the heart of it are master regulator genes and signaling proteins. These play crucial roles in determining wing tissue identity and in driving the wing's development forward. Notice the theme returning. A master regulator gene does not act alone, it works through, and alongside, signaling proteins, so that the right tissue knows it should become wing and then develops accordingly. This is gene interaction made visible as anatomy. The same logic we met in coat color and flower color, genes depending on and cooperating with one another, here sculpts a functioning wing out of a field of cells. If genes must act in the right place and time, the cell needs control, and that brings us to our third theme, the regulation of gene expression. Gene expression is tightly controlled so the correct amount of protein is produced at the right time and place, meeting the cell's needs for specific proteins. Crucially, this control can occur at multiple levels. The first and most prominent is transcriptional regulation, the primary mechanism by which gene expression is controlled. It works through transcription factors that bind to DNA and can either enhance or inhibit the start of transcription. This is where positive and negative control live. Positive regulation uses activators that enhance gene expression, negative regulation uses repressors that inhibit it. The lac operon is the classic example of negative regulation, where the loci repressor controls the genes involved in lactose utilization. Transcription is only the first checkpoint. Once an RNA is made, post-transcriptional regulation takes over, acting after transcription through processes such as messenger RNA processing, transport, and stability, all of which affect the final amount of protein produced. That processing includes splicing, capping, and polyadenylation, and the journey of the messenger RNA out of the nucleus to the cytoplasm is itself regulated. Then comes translational regulation, which controls the rate at which messenger RNA is translated into protein, and this can occur at the initiation, elongation, or termination stages of translation. Even after a protein is built, control continues. Post-translational regulation acts after protein synthesis through modifications such as phosphorylation, acetylation, and ubiquitination, which can alter a protein's activity, its stability, and where it is located in the cell. Control at every stage. Step back and ask why a cell bothers with all these checkpoints. The answer is timing and location. Temporal regulation ensures that genes are expressed at the right time during development, while spatial regulation ensures that genes are expressed in specific cell types or tissues, and both are critical for the proper functioning of multicellular organisms. Developmental signals and cell-specific transcription factors help steer this, and the lac operon shows the same idea responding to the environment, switching genes on in response to a signal like lactose availability. So bring the whole episode together. Linked genes are inherited together and mapped by recombination. Multiple genes interact, additively or epistatically, to build a single trait. And every gene is regulated, at the right time, in the right place, through every stage from DNA to finished protein. That is how genes truly behave together. See you next time.